It's my pleasure to speak to you today about precision genomics and immunotherapy in the cancer field and beyond. These are my disclosures, and I want to start with talking about my experience that has really informed my thinking on precision medicine in the clinic. The most recent experience is as the director for the Center for Personalized Cancer Therapy at UCSD Moore's Cancer Center, where there was a real emphasis of bringing the basic science and the clinical care together to help patients with cancer. And before that, I was the founder and chair at MD Anderson Cancer Center of a very large clinical trials department that did early phase clinical trials, meaning that these were cancer patients that had really failed all therapy. And this was back circa 2007 when we started using precision medicine in its earliest form. And it really gave us a glimpse into the potential for this to really transform the care for patients with cancer. So I have several take-home points, and the main being that precision medicine is all about getting the right drug to the right patient at the right time. And the main take-home points is that first, at the genomic level, every metastatic tumor is unique and complex, what I'm going to refer to as malignant snowflakes. I live in San Diego, but I'm from Canada, so I know a little bit about snowflakes. And every snowflake, its crystal structure is very complicated, and every snowflake is unique. And we really think metastatic tumors are at the genomic level similar in that way. The second point is that metastatic disease requires customized, individualized combination treatments, not single agents. Third point is that the pillars of precision medicine are genomics and immunotherapy, and people think of them separately, but they are really married to each other. And the final point is that immunotherapy is best suited for genomically complicated tumors. Some of the tumors that we previously thought were really untouchable, that's where immunotherapy fits in best. So genomic technology has undergone some amazing changes, and this is what has really been able to feed this revolution in precision medicine. The first human genome was sequenced about 2003, and it took 13 years to sequence a single genome. Now we can sequence it in a day. At the time, it cost $2.7 billion to sequence a genome. By about 2007, it was down to $100 million per person, which was still a little bit steep for insurance or for research labs. But now we can sequence a genome for $1,000, and that makes the technology usable in the patient setting. So to me, this is breathtaking progress, unparalleled in human history, that has really fed our ability to perform precision medicine in the clinic. What this has led to is a very remarkable evolution in clinical trial design. So classically, we have thought about cancers as where did the cancer come from? If it came from the lung, it's lung cancer. If it came from the breast, it's breast cancer. If it came from the colon, it's colon cancer. And we thought of each of these as one disease. But with genomic sequencing, we began to realize that, as an example, lung cancer was actually many subsets of disease defined by their genomic alteration. But furthermore, we also came to realize that the same genomic alteration could exist in many different types of cancer. You might find the same alteration in colon cancer, in liver cancer, in brain tumors, in lung cancer. And furthermore, that we could treat patients based on that genomic alteration, regardless of which organ the cancer originated in. And this has really led to some transformative changes in clinical trial design. So our standard model was randomized clinical trials of unselected patients where we would see a small number of patients respond. The new model is small patient populations, highly selected to match with the drugs that they're given, and where we expect to see much higher response rates. So this has led to a new era that was really almost unimaginable even a decade ago. And that is what we call tissue agnostic approvals driven by genomics. And what that means is the FDA approving drugs based on the genomic alteration, and the organ of origin doesn't actually matter. These are approvals across solid tumors, for example. The first of these was on May 23rd, 2017, when the FDA approved an immunotherapy, pembrolizumab, for all solid tumors based on whether or not they had a mismatch repair gene defect. And there were three remarkable aspects of this approval. The first two, many people know. The third, which is probably the most remarkable, is probably the least well known. So the first was, as I mentioned, it was a tissue agnostic approval, was for all solid tumors. Didn't matter if you had lung or colon or breast cancer, etc. The second was that it was approval based on a genomic marker, and it was approval of an immunotherapy based on a genomic marker. And the third aspect of this approval, which to me is the most remarkable, was that in part it was based on retrospective real-world data, not just on prospective clinical trials. And since then, the FDA has approved another indication for a drug, palbociclib, that is entirely based on real-world data. And with all of our computerized records, this really makes possible very rapid approvals as we look into our databases and look at efficacy based on real-world data. But that's just the beginning. Since then, the FDA has approved a drug called larotrectinib, specifically for patients with a genomic aberration of NTRK fusions. And this is another tissue agnostic approval in adults and children with solid tumors, doesn't matter what their tumor is. And then another approval for entrectinib, another drug for NTRK fusions, again, tissue agnostic. And then most recently, pembrolizumab, again, immunotherapy, approval based on tumor mutational burden high across solid tumors. And this most recent approval was in June of 2020. So I want to step back a bit and talk about genomics and immunotherapy and the relationship between them. So the pillars of precision and personalized medicine, to me, are genomics and immunotherapy. And many people think of them as separate entities, but they're really a couple and they're married to each other. And what do I mean by that? The genomics is often the determinant of whether or not immunotherapy works. And the reason for that is that when we give immunotherapy, specifically immune checkpoint blockade, what we are doing is we are reawakening the immune system. But the reawakened immune system still has to recognize the tumor. And the more mutated the tumor, the better the response to immunotherapy. And this makes sense because the more mutated the tumor, the bigger the difference between the tumor and the normal tissue, and that makes it easier for the reawakened immune cells to recognize that the tumor is abnormal and is different from the normal cells and to eradicate it. So what we found is that the response rate for immunotherapy for low mutational burden was 4%, but when you get to very high mutational burden, the response rates goes up to 67%. While at UCSD, one of the central features of our program was a molecular tumor board. And this molecular tumor board brought together physicians of different specialties, medical oncologists, surgical oncologists, radiation therapists, radiologists, pathologists, and so forth, with our basic scientists, with our computer specialists. And we did molecular profiling, clinical grade, on about 21,000 patients. And the idea here was to present patients and to make decisions and give them therapy. So this was not just about doing research, divorced from patient care, but doing research, but also informing better patient care. So I'll give you some examples of some of the work that we did. And I want to start with individual patients because I think that they illustrate some of the potential. This is a patient who with metastatic basal cell carcinoma, which is a very rare tumor. I actually saw him in a clinic about 6 weeks ago. At the time we treated him, which was October 2015, he was 55 years old. He'd already failed 6 different therapies. He was in a wheelchair, not doing well. He had brain, liver, and bone metastases. And he'd failed the FDA-approved therapies, which are targeted therapies like vismodegib and sunitinib, which target the classic alteration, the classic genomic alteration in these patients. And that's a patch-1 alteration or hedgehog. But he didn't respond to those therapies at all. And so what would you most likely do next for this patient? I think a lot of people would say, well, send to hospice might be appropriate. But because we were doing a lot of genomic profiling, that's what we did. And what we found was very interesting. The first thing was he had the target. He had the patch-1 target, which mediates response to this vismodegib and sunitinib, the therapies he had failed. So why did he fail those therapies? Well, I think it's obvious just by looking at his genomic profile. He had multiple other alterations that were driving the disease. So targeting one of them was just not good enough. And very interestingly, he had a very high tumor mutational burden. Anything over 50 we consider very high. And he also had a marker that we know now and we've published is associated with response to immunotherapy called PD-L1 amplification. So we treated him with nivolumab, which is an immune checkpoint inhibitor. This is his liver. This is a scan, and this is his liver before treatment. And this is 2 months after treatment. And fortunately, he went into complete remission, which is ongoing now at 5.5 years. As an oncologist, I'm a bit hesitant to call anybody cured, but I think that he may be cured of his disease. And again, these types of responses were unimaginable a decade ago. But I also want to talk about some of the downsides of immunotherapy and how genomics can help us with that, because it's not all good, even though we all believe immunotherapy is some of the most important types of new treatment that we've discovered. Some patients do poorly with it, and there's a phenomenon called hyperprogression. And what hyperprogression is, when you give immunotherapy and patients progress in an accelerated fashion. In fact, it's almost as if you've poured gasoline on a fire. So this is an example of a woman who unfortunately experienced hyperprogression. 44 years old, triple negative breast cancer. This was her scan 2 months before she received immunotherapy. This was her scan right before immunotherapy. And you can see the tumor hasn't changed much. And then 6 weeks after immunotherapy, her tumor explodes. And one thing to note is that in her genomic profile, she has an MDM2 amplification. And what we found is that there are 2 genomic alterations, EGFR alterations, and especially MDM2 amplifications that can predict for hyperprogression or accelerated progression. So the important point here is that next generation sequencing can help define not only who to treat, but who not to treat with specific therapies. And that's probably who not to treat is probably as important as who to treat. So I'm going to give you another patient. This is a patient of mine, a patient with what's called a high-grade neuroendocrine cervical cancer. This was a 49-year-old woman that came from Saudi Arabia, and she came to see me in January of 2016. She had been treated with surgery, chemotherapy, and radiation therapy, and she had extremely advanced disease. She's had huge abdominal tumor, looked like she was 7 months pregnant, but it was all cancer. She had impending bowel obstruction, impending kidney obstruction, and she unfortunately did not have very long to live. So again, the question is, what do you most likely do for this patient? And sent to hospice might be a perfectly reasonable answer. We felt bad that she had traveled so far, so we wanted to do comprehensive genomic profiling on her. But we had a problem, and that was because she only had a week or two left to live, doing a tissue biopsy and getting the profiling often takes a month. So that was just too long a period of time. And that's where new technology comes into play. So what we can now do is liquid biopsies, and these are interrogating the blood. And all we need is a blood specimen, and we can do gene sequencing on the blood specimen by extracting shed DNA that comes from the cancer and is shed into the blood. And of course, this is much faster. And this is what we found on this patient. The average patient, when we look at the blood, has about 3 or 4 alterations, and she had over 20 alterations. So at the time, we took a quick guess and thought she may have hypermutated DNA, and that means a high mutational burden. And so we treated her again with immunotherapy with checkpoint blockade. And this is her tumor before therapy. This is 8 months after therapy. And my last contact with her, she did return to Saudi Arabia, was 3.5 years after treatment. And she had an ongoing complete remission at that point. Again, a remarkable response that was really unimaginable just a few years ago. Once we treated her, however, we did do the tissue biopsy to try and figure out more information about what was going on with her. And sure enough, she had a lot of genomic alterations in the tissue genomic sequencing. The average patient has about 4 genomic alterations. I think she had 23. She had a very high mutational burden, over 50, it was 53. And it turned out that she had microsatellite unstable disease and a mismatch repair gene defect, which we now know is an indication for immunotherapy. And we also now know that the blood tumor mutational burden can be usable to determine therapy for patients. So an important point here, cutting-edge technologies are needed for complicated cancer, and blood-based tumor mutational burden and microsatellite status is now available in a clinical grade form and usable really by any oncologist. So this was all very exciting, but as sequencing became more powerful and gave us more information, originally we were very excited about that. And then we realized that it was telling us something that really complicated our lives. And that's, what if every patient with metastatic disease is different? And that brings us back to the snowflakes. As you can see from their crystal structure, each of the snowflake is complicated. Each snowflake is different from every other snowflake. And that's what we've really discovered is true for metastatic tumors. And we've sequenced, as I said, over 20,000 patients. But I'm just giving you this slide because this is what fits on a slide, and I want to use it as an example. This is sequencing of breast cancer, and I want to use these two patients. Both of them have something called ERB2 amplification, which leads to HER2 overexpression. And this is a known treatment that's FDA approved, which are HER2 targeting agents. But if you look at their sequence, you can see that even though these two patients have something in common, the rest of the molecular portfolio is distinct from each other. And it's pretty complicated. There's more than one alteration. And so this idea that we treat patients that have something in common with the same regimen doesn't really work if we think about what the genomics is telling us. Yes, these patients both have something in common and need one of the same drug, but for the rest of the abnormalities in their genomic portfolio, they need an individualized combination of therapy. And that's not something that we've done before. So instead of using a consistent drug between patients, even in the presence of different molecular profiles, which is the traditional way, we wanted to use a consistent strategy, which is molecular matching, but allow different drugs matched or customized to each patient. So here is the idea. This would be a patient-centric trial where every patient is considered as an individual. And even if both patients, patient 1 and 2 as examples, have marker A in common, the rest of their portfolio is different. So patient A would get drug A, B, and C, and then patient 2 would get drug A, E, and F, an individualized combination. And that gave birth to a trial that we called PREDICT for profile prospective investigation of profile-related evidence determining individualized cancer therapy. And the idea here, we initiated this in 2015. The idea was to give customized combinations. And there was a second part of this protocol where we treated newly diagnosed patients with lethal malignancies, trying to treat them earlier before their disease really became resistant. And we published the first part of this in Nature Medicine in April 2019. And this is what we found. So the first finding is that matching patients to drugs based on genomics is not a yes or no answer. It's the degree of matching that really counts. So a patient that has 10 alterations and is matched to only one drug gets a very low matching score of 10%. If you have 10 alterations, it's really important to match many of those alterations, not just one of them. And patients with a high matching score, their response rates were about 45%, keeping in mind that these patients have refractory disease and have failed to respond to FDA-approved therapies. And then in contrast to the 45% for high matching score, patients with a low matching score or unmatched had a response rate of 16%. And then when we look at survival, it's really remarkably different. The patients with a high matching score at 2 years, their survival is about 60%. The patients with a low matching score who had very low matching or no matching, their 2-year survival is only about 20%. So the bottom line is that the high matching score translates into significantly better response rate, better survival, and better progression-free survival, which I didn't show. So this is an example of some of our individualized therapies. And this is a 62-year-old man with poorly differentiated carcinoma of unknown primary, meaning we don't know where the cancer came from. But we do know the genomic alterations. And the patient had an atypical KRAS alteration, which, however, is deleterious. And KRAS activates the MEK pathway, so we gave the patient a MEK inhibitor called trametinib. The patient also had an ARID1A alteration, RID1A is a chromatin remodeling gene that does many things, but there's data supporting that it may be sensitive to PARP inhibitors. So we gave a combination of both drugs. You can see the dramatic response that this patient had with therapy ongoing at 15 months. So I'll just mention some other important initiatives that I'm involved with. I've worked with the Worldwide Innovative Network for Personalized Cancer Therapy for over a decade. This is dedicated to the global delivery of precision medicine. And the idea that genomics is only the tip of the iceberg. There's transcriptomics, proteomics, immunomics, epigenetic changes, and so forth. And as a first effort, WIN has been looking at the importance of the transcriptome or the RNA expression in patients. And the first paper published by WIN, again in Nature Medicine in April 2019, was a international trial of precision medicine that for the first time incorporated not just genomics, but also transcriptomics. 303 patients were enrolled, and the trial showed that transcriptomic understanding remarkably increases the number of patients that can be matched to therapy and how well they can be matched to therapy. And there's another new type of trial that I'm very excited about. And a great example is TAPROOT, which is a master observational trial. I mentioned early on the importance of real-world data. Well, this is real-world data, but in a structured form. An observational trial taking place nationally, which will collect data structured in order to better understand new therapies for patients with cancer. And then the last couple of things I want to mention here is the host. I've talked about a lot about the genomics of the tumor, but we've paid less attention, need to pay more attention about the host, because the host informs toxicity, immunity, and microenvironments. And this person, who actually isn't a patient, but obviously a chain smoker, pretty robust and in your face, celebrating her 100th birthday. And, the question is this good luck or is this genomics? And I don't know the answer, but I'm willing to bet that it's her genomics that determined that she would stay in good shape, even though that she's a chain smoker. And extrapolating from there, I think genomics can tell us a lot about how a host will respond to an individual therapy. It's an area that we need to pay a lot more attention to. And then the last thing I want to mention, which has just fascinated me, is how we take this beyond cancer. And I'm going to give you one example, but there are a lot. The FGFR3 gene is involved in bladder cancers, and it activates FGFR. And there are therapies, targeted drugs, that can be used to inhibit it and get responses in bladder cancer. The exact same gene in the germline form, in the hereditary form, causes achondroplastic dwarfism. And so the question is, could we use the drugs that inhibit the activity of this gene in order to normalize the phenotype of somebody born with achondroplastic dwarfism? I don't know the answer to that, but it has been tried in mice with achondroplasia, and at least in the mouse models, it seems to be effective. So I will finish by giving a special thanks to the precision medicine team, and especially to our patients, and I will be happy to entertain questions. Thank you.
Razelle, one of the things that it just sort of seems to me to be a theme throughout what you just talked about is this is how promising all of this sounds and the directions we're going in. But what's in the back of my mind is how do you frame this in your own mind in terms of what you are talking to patients about? Do you see what you're doing at this point as simply a treatment option, or is this all an ongoing research project, or is it both? How do you frame that?
So I think that's a really interesting question. And really, for me, it's become a treatment option. But I think for most of the world, it's really a research question. I've been doing this now, this field for over a decade, sort of an early adopter, and I can see the power and how it makes sense. So the bottom line to me is that we now have tools that we did not have 20 years ago. And we can understand what is wrong with an individual patient's tumor. There's no more guessing. We don't have to have guessing games. Our science isn't perfect, but before we used to treat on the basis of population averages. We'd have 50 patients with colorectal cancer. We'd put them on a trial. 20% would respond, and we would give that therapy to everybody. And again, every patient that came in, 20% would respond, just like in the clinical trial. 80% would not. We didn't know who would respond, who wouldn't. We just didn't have those types of tools. Now we have much better tools. We're not perfect at this by any means, but we're way well on the road. And when a patient comes into me, I don't care if they have the rarest genomic profile you could ever imagine. They may be 1 in 1,000, but that really doesn't matter. They don't need to be treated as a population average. They need to be treated as that individual with that profile. And so to me, this makes complete sense as a treatment option. But I think we're still on the road for this type of thing being adopted. And we do it under the auspices of clinical trials. That's how we've done it at UCSD. That's how we've done it at WIN. And we now have national trials that I'm leading and other people are leading. So we're doing it under the auspices of clinical trials. I think that within 5 years, I hope that this is the way everybody is going to practice.
Great. Well, thank you. And so, Patricia, why don't we take your question and get the ball rolling here?
Yes, actually, doctor addressed that a little bit. I'm in remission. I was diagnosed in 2013 and continue to be a patient at Moore's. It's interesting to me that the biology now is being used for the treatment. And so I wanted to ask if she could maybe even predict, since there's been such a dramatic change just since 2013 for me, whether this is going to be applied relatively soon to tumors that have not metastasized.
First of all, I'm very happy that you remain in remission now so many years later. And that's obviously a very good sign. And I think you've asked a key question. And in fact, it has been part of the clinical trial that we did. So we did the clinical trial that I spoke about, I Predict. We initially looked at patients with who had failed all therapies, and they had metastatic disease that had been through a lot. And we know those are the toughest tumors because the therapies, if they're not successful, they drive resistance. And we also know that if you can give the therapy earlier in the course of the disease, the tumor is not as hardy, it's not as resistant. So part of that trial was to give newly diagnosed patients with lethal cancer. So they had to have an expected mortality of 50%. of the patients would die at 2 years before we could do this, because it was considered a research question. And to give them right up front, as soon as they were diagnosed with metastatic disease, give them these customized matched combinations of therapy. We haven't yet published the results, but we've submitted them. And I think it's very close to acceptance. And the bottom line is that what we expected was true, that if we give the patients the therapy earlier, they do even better. Right. And then the next question, so you have to do this gradually, is what if we go even earlier? Why wait for the patient to have metastatic disease? If we can impact these really horrible tumors that are metastatic and there's a lot of tumor burden, what would happen when the patient comes in with a 1 centimeter tumor, with a tiny little tumor? Could we completely eradicate it with understanding the biology, looking at the genomics, looking at the immune environment, giving them the right therapy, and just get rid of that 1 centimeter tumor without having the patient go through surgery that might be very difficult, or radiation? So I think that's the next question I'm very excited about. It's much more difficult to do a research question in that setting, because a lot of those patients are already curable, maybe curable with some tough therapies, but still curable. So that becomes an ethics question. When can you move it up even further?
That's actually a really good point. And actually, it points to one of the questions I wanted to ask, get on the table is, how far can this reasonably go? I mean, I was really struck by your question, what if every patient is different? And I'm wondering, by the way, as a sidebar, maybe that should be what if every tumor is different, right? Because it's not really the patient's DNA so much as the tumor. And I guess it's even feasible, I presume, that somebody could have 2 different kinds of tumors in their body. And so you might have, so it's really the tumor differences. But how far can this go? I mean, do you see that the trajectory right now is that we will simply get better and better in an incremental way and eventually be able to target any cancer for any individual? Is that where this is going, or is there going to be or do you anticipate there's going to be some point at which you have reached a maximum benefit from this approach?
No, I think it's going to get better and better. And in fact, when you look at what's happened to science, especially medicine, all the predictions of the trajectories of the development of science and knowledge have been off. But they've been off because even the most positive predictions of the trajectories have underestimated how fast things have moved along. And so I think it's going to go get better and better. I don't think I don't really think that there's a plateau. I would like to see cancer eradicated in my lifetime, and I'm not supposed to hype anything, but I don't want to get in trouble with any colleagues, but I actually genuinely believe that we're going to do this.
Yeah. So, well, this reminds me of a conversation we had back in 2014, I think, when you served as moderator for Patrick Soon-Shiong, who was one of the speakers in the Ethics Center's Cancer Series. And you might remember, he was showing some remarkable, promising results with pancreatic cancer, where he showed a patient who, by all rights, from what everybody else has seen, would have died in a short period of time, and their tumor load seemed to have disappeared. It looked so promising. What is the trajectory there? How far are we from success? Is that sort of, at least for the time being, plateaued?
So pancreatic cancer, that's been a difficult one to crack. I hear what I've heard many times in my career, that it's never going to be treatable. And I can't tell you how many times I've heard that. And every single time, not every single time because pancreatic cancer, but many times that has not been true. Just to give you an example, a disease called GIST that at one point, it was worse than pancreatic cancer. The response rates to any chemotherapy were 0. And then it was discovered that this tumor was driven by KIT mutations. And there was this new drug called imatinib that is also used for chronic myelogenous leukemia. And there was a single patient published in New England Journal of Medicine. This patient was published 6 months after a premier review article, which said that there was no way to treat this cancer. And this single patient, they were able to show that when you gave this drug, imatinib, which is FDA approved now, the tumor turned off its metabolism within 24 hours. So science is really an amazing thing. And so that's just one example of a tumor that's considered completely untreatable with 0% response rate, worse than pancreatic, because pancreatic cancers actually have responses, that was completely transformed with one patient. And now this is a very treatable cancer. I've heard this already several times. I absolutely believe in the science behind what we're doing, and that new discoveries will make even the most difficult cancers treatable.
Yeah, so that leads to another question that I have that leads to a question that's in the chat. So first, having also at the same time as Patricia, having been diagnosed with cancer in 2013, I'm directly personally familiar with that tension that exists between a physician or researcher, depending on how you see yourself at any given point, noting that there has been some wonderful success, that possibility, that hope that helps somebody keep going, and balancing that with the likelihood, where in some cases the likelihood of success is right now, unfortunately, low for many cancers. So I don't know to what extent you could talk about that tension between hope and likelihood. I mean, how do you deal with that? You don't want to promise the moon because you can't give the moon, but you also don't want to tell somebody there's no hope.
Yeah, I think you're addressing a really important question. There's a lot of controversy about that. And I consider myself a physician first and foremost. And the research is all about the patient. The whole purpose of the research is to help that patient get better. Clinical research that does not benefit patients, and there is that type of clinical research, I've never participated in that. It's just my strong belief that I want to participate in research that I believe can help patients. So I genuinely think that most patients are not that naive. Most patients that come in with metastatic aggressive cancers, some of them may be in a bit of denial for what they say, either for their families or to give themselves hope. But I think pretty much everybody in America knows that if you have metastatic pancreatic cancer, you're in big trouble. And so I think, as personally, I try to balance it out and give a realistic expectation. And my own finding is, especially that patients who seek out clinical trials, I don't think that they're unrealistic. I think that just the act of trying improves their life, because there's many patients that don't seek out clinical trials. They say, I'm done. I, want to stay home. I want to have time with my family. But I find that the patients, both at MD Anderson and at UCSD, that seek out these clinical trials, I've had many patients that have said to me, don't give up on me. Even if it's not going to work, I want to know that I'm trying. And so I think there are ways to balance. And I think it's important to know that for patients who seek out these trials, just that you're trying, that gives them a better quality of life.
Great, thank you. So one of the questions is about the regulatory environment that you're in for this to happen. So somebody asks, are you required to obtain FDA approval for the use of these drugs on specific patients? Or is there some sort of global approval for the approach?
Right. So it depends on what you're doing. If the drug is called experimental, which means it has not been FDA approved, or if any part of the combination that you give is experimental, you have to obtain an FDA, you have to obtain an IND, and you have to be doing it under the auspices of a clinical trial. And the FDA has to approve what you're doing with that drug. But there's another class of drugs that are FDA approved, but we use them what I will call off-label. In other words, the drug may be approved for colorectal cancer, and we're going to use it for breast cancer. That has been always legal for us to do that in the United States. And indeed, oncologists, about 40%, even if you go into a community practice, about 40% of the drug usage for cancer patients is what's called off-label, where the oncologist gets, I guess you could say, creative with different drugs. So some of what we do is off-label. And I think the difference between what we do and the community oncologist does is that we're very focused on understanding the biology of an individual patient and giving them the right drug for that situation. So the off-label use of drug is not limited by the FDA. It is limited by the expense of the drug, and if you can find ways to pay for it. And we really set up a really good system at UCSD where we're able to obtain drugs because there's many mechanisms in the United States to obtain these drugs without saddling the patients with the financial burden of them.
Yeah, actually, that sort of jumps ahead to a question I was going to ask later, but what is the insurance environment now for paying for drugs that have not yet been approved to target the actual tumor rather than the location of the tumor? So
that is off-label use of drugs.
Would insurance companies cover that off-label use?
So in general, government insurance will not. But private insurance, for some reason, in my experience, about 40% of the time will cover it. For no good ascertainable reason that I can see, and then there are other mechanisms to also obtain many of these drugs for patients. Many of the companies have what's called a compassionate use of the drug, where you can get the drug, the company will give you the drug without asking for payment. And there's other mechanisms, foundations and so forth to get these, to get drugs for patients. I don't know that that's scalable for the world, but I've always viewed our role as showing this works. And then the FDA and the system needs to figure out how to scale it once we can show it works.
I have to say what you've just said is remarkable compared to the kinds of discussions we've heard in these programs in the past. There was a presumption or worry that insurance companies would almost never be willing to cover something like this. And the fact that quite a high percentage do is very good news. And I think there'd be a worry that any kind of use like this might be seen as being in it, being research, and so there's not going to be coverage for it. And that's very nice to hear.
So I do want to make some comment about this. So we do it under a protocol, under a clinical trial. And many insurance companies will not cover it. So when I said 40%, that's definitely not the majority.
Oh, yes, but it's still a high percentage.
It's still higher than people. So the rumor that there's no coverage is not correct. Insurance companies will deny it, and in my opinion, have the right to deny it because it's not standard. But there's something else that insurance companies, and actually the government insurance, Medicare, will cover many things in a clinical trial, and that's by law. So many aspects of a clinical trial, even if you have, and I'll give you an example, if you have an experimental drug, they won't cover the experimental drug, but they will cover the infusion costs of that experimental drug. And they will cover the blood work to monitor the side effects for that experimental drug. And that was a law that was created actually by Bill Clinton many years ago. And Medicare covers it, government insurance covers it, and many of the private insurances, if not most, follow suit and cover many aspects of clinical trials.
So I'm going to move to a question that's in the chat. And this is, I think, something that you alluded to more than talking a lot about in your talk, but you were pointing out that you have both the genomic and the immunotherapy approaches one can combine. So the question is, can the combined application of CAR T therapies, along with the other genetic matching immunotherapies, find a useful approach to metastatic solid tumors?
So I think that is a really good question, but I think the first part of that we need to address is CAR T therapies in solid tumors. So CAR T therapies have been amazing in some hematologic malignancies. And for some lymphomas and leukemias, patients that you would never think had any hope. They get their CAR T therapy and they appear to be cured. It's really amazing. But the thing about CAR T therapy is that it's really a precision medicine therapy. It's a very personalized therapy. The T cells have been engineered to recognize the tumor. They recognize part of what's on the cell surface of the tumor. And they've been, these T cells, their job is to kill things that don't belong in your body, whether it's bacteria or viruses or cancer. And these are specifically engineered T cells, so they already know how to kill things, but they didn't know how to recognize the tumor before. But they've been engineered with amazing science, I think, so that they recognize the tumor and they can therefore kill it. So this is like almost the ultimate personalized therapy.
Yeah, and I just want to underline that the goal, or the approach is not that they have developed T cells that can recognize any tumor. They have been designed to recognize the specific tumor in this specific patient. So that's why you're saying it's highly individualized.
Well, it may be the specific tumor in the specific patient, or it may be something that's on most tumors that are a specific type of tumor. So there might be a specific lymphoma that each of them have something on their surface. And so whatever you engineer the T cell, and it's good for most patients with that lymphoma because that is the marker that the lymphoma carries. Or the other way to develop them is to individualize the CAR T cell to recognize one person's tumor.
And that's when it becomes highly personalized, I guess.
Right.
Yeah, makes sense. So I have a question about the context in which you're doing this work. So in a normal, a regular, typical clinical trial of drugs and interventions, usually you need lots of people in the study in order to get enough data to know what the relative chances are that this actually works. But in this case, you really are working in a world where in some ways it's always an N of one. It's one person per trial. So how do you think about that from a research perspective?
Right. How do you know it works, right?
And what does that mean to work? Yes, exactly.
Yeah. So if everybody gets a different therapy, how do you know it works? So I'm going to try to articulate it. So we are not evaluating the efficacy of a specific therapy in a specific disease, because to do that, you need 20 to 1,000 patients that get the same therapy. And all our patients get a different therapy, but there's still something that we can evaluate, and that's how we did the studies. We can evaluate our strategy. We can evaluate the strength of our strategy. So what we do is we calculate how well the patient was matched with their therapy, and we give it a matching score. So we calculate how well we were able to carry out what we wanted to carry out. And then we ask the question, how effective was this strategy? If a higher matching score correlates with a better outcome, our strategy is effective. And that's exactly what happened. So I showed you in the published study in Nature Medicine that the patients with a high matching score, which was over 50%, their 2-year survival was about 60%. The patients with the low matching score or zero matching score, but also anybody less than 50%, their 2-year survival was about 20%. The difference between a 2-year survival of 60% and 20% is quite dramatic. But again, we were evaluating the strategy. Every patient gets a different therapy. We're not evaluating individual therapies. We're evaluating the algorithm of us figuring out how to give these patients drugs.
So in this context of this kind of research where you're looking at a strategy instead of an individual drug or individual treatment, which makes a lot of sense, how do you approach the informed consent? I mean, so the people that we're talking about are in a state where probably everything else has, they've tried, and this is the last resort. And so in certain senses, they are desperate. So how do you convey to somebody in that circumstance the nature of this being experimental and not clinical? What are the words you use, the approaches you use to convey that in informed consent?
Yeah, so I think the first thing is that just by presenting an informed consent document, we're telling them that this is part of a clinical trial. And therefore, it's still a research question. And, I try to be pretty transparent with patients, although a lot of them have, done their reading or looked at the videos on the, on the internet, or even looked up my papers. I'm surprised how many people come in and have looked at their genomics and tell me what I'm going to prescribe. And they actually have it pretty much right. And not all the time, but and we tell them that everybody's going to get a different therapy, that we're going to evaluate the efficacy of our strategy, and that the early evidence and what we believe is that we're weighing the dice in their favor. By understanding the biology of the tumor and trying to give them the best therapy. And at this point, we also tell them that we have publications that support that. But I also always tell patients that the science is not perfect, that unfortunately many patients do not respond. Some patients will have side effects, maybe significant side effects. And so we cannot guarantee the outcome. The only thing that we can guarantee is that we will try our best.
I have a question for you that goes back to my own treatment. And that is that I realized as I watched the process that if my physician told me the likelihood of how bad the side effects could be and how bad some of them were, I might have been more reluctant to go through the treatment. In my case, I was kind of on a borderline between definitely have to get treatment and definitely don't. So as a result, it was more of a choice. And I've realized in retrospect that in some ways, if they think the best thing is for me to have the treatment, then the best thing is for them to not push too hard on how bad the side effects are. Have you dealt with that sort of a question when you're thinking about how to talk to a patient?
So I tend to be more upfront with patients. And I tend to, but one of the issues is it may be that 90% of patients don't experience bad side effects and 10% do. But I try to give patients a really realistic point of view of what their side effects will be. And also why, at the same time, telling them why I think they should be doing whatever I think they should be doing. So I don't know if your physician minimized it or maybe you were more sensitive to the drugs than a lot of other patients, because it's really hard sometimes. Sometimes we use drugs and really most patients sail through it, and then you give it. And, I always tell a patient that I can't tell you how it's going to be for you as an individual. And then the person in front of me, has horrible side effects. And the opposite occurs too. I've told patients that this is a really tough therapy and most people have problems with it, and I'll go through all the side effects, but this is what you need to do, or in my view, and this is why. And then the patient has almost no side effects. So sometimes it's not just the physician minimizing. It might be, but it might be also where you are on that spectrum of vulnerability.
Good, thank you. So, because we're all scientists here, everybody, if you showed up today, you are by definition a scientist because you're trying to figure out how things work and how to do them better. I'm going to try again to use breakout rooms, which we've only done a few times here. And what I'd like to propose is that we're going to ask your help, the participants in today's program, your help in thinking about what do we need to convey to somebody who might be in one of these research studies for proper informed consent? What are the most important things you would like us to know in order to make that decision? So we're going to do a whirlwind tour of the groups. So room 1, I hope you remember which room you were in. Can one of you tell us maybe the most important things you thought should be included in the informed consent process?
So my question is about how you convey risk to patients in a way that they can grasp. I have a math background, and I feel like observing the COVID pandemic over the last year that not everybody has the best grasp of statistics.
Good point. So actually, why don't we hand that to Razelle? So probably one of the questions that's going to come up is probability. You tell somebody the probability that your cancer will kill you in X number of months is this. And how do people understand those numbers?
So again, I tend to be very concrete and very very honest about it. It's actually a very difficult conversation to have, and I think that many physicians have trouble with it. But I just tell people exactly when they ask me, what is my chance of being alive a year from now? It may be 90% or it may be 20%. And if it's 20%, I will say it's 20%. And that I find that difficult as well because, it takes people's breath away. I do always add that there is variability and everybody is different. And so you can't, you're not a statistic, but you ask me a question and I'm giving you the statistical answer. And I really do think people understand, like I have a 20% chance of being alive at 1 year. That's really bad. Or I have a 90% chance of being alive at 1 year. It's not perfect, but it's pretty good.
So your sense in response to Elizabeth is that people do seem to have a sufficient understanding of probability and what that means.
I think that if you keep it simple, people really understand it. And I do think a very common question for patients is, how long am I going to live? And I give them, if I think it's 6 months, I will say, I think it's the average patient is 6 months. I don't know if you're average or not. That would be my answer. I give simple, straightforward answers.
Okay, good. Thanks. So let's go to room 2. One of you can tell us what you came up with.
Hi, we talking about, again, those risks, which can be small but serious, like dying. So, or whatever else, neurological sort of things, pain, gastrointestinal things, osteoporosis, and sort of saying these are all possible downsides. Okay, that's it.
So convey those risks.
Yeah, so I think, again, we try to give patients a realistic outline of the risks. And if the consent forms often have the risks extensively outlined. Unfortunately, I think that when you have 10 pages of risks, it's almost not useful. So the patient will, sign the consent, and I don't know that they read all 10 pages or absorb that. And really what they want to know, in my experience, is what is my experience? And I try to summarize that and to answer their questions transparently.
Good, thank you. Kamudra, did you have something you wanted to add? I saw your hand up a moment ago.
Yes, well, it's just to kind of add up to what we discussed. It is true that there are so many side effects it's possible, and the consent form, it can be long, and if I were a patient, I would read those consent form rather than signing. But most likely, as from a patient point of view, I guess the best way is to be able to answer their question from an expert point of view, and I would really agree with what Razelle said, the honest, the better, the simplest answer, it's the better for the patient. So I would say that would be the best way to convey consent from patients.
Okay, good, thank you.
I agree. And they have the consent form so they can go through every one of those side effects. And there are people who want to do that, but most patients don't. Most patients want to trust their physician to give them a view of what is likely to happen, with an understanding that there are outliers, both in the good sense, outliers that do much better, and outliers in the bad sense, people who unexpectedly do very poorly.
Thank you. So room 5, Carol or Nancy, can you give a summary for your room?
Hi, this is Nancy. We understood the assignment to be to make a list of things that you want to see on an informed consent document. So Carol started rattling them off and I started writing them down. And it's a very long list, but I've been checking off the ones that have already been discussed.
Maybe mention a couple of ones that are close to the top of your list then, that you think would be relevant to add?
As far as our list is concerned, the highest remaining is what is the procedure that I'm agreeing to, and what am I expected to do to participate in this? And I think that goes along with the tests that will be required of me if I agree to do this. Will they be blood tests, biopsies, that type of thing, and the intervals between the test and things like that. I think what remains on our list is, can I change my mind during the clinical trial? And finally, will my cancer be most likely to kill me, or will this clinical trial be likely to kill me? That kind of thing. Is the clinical trial so risky that it may kill me even though my cancer hasn't had a chance to yet?
So I want to address all of those things are in the consent, and actually the consents usually have a detailed step by step when you have to give blood, how much blood, how often you have to come in, and so forth. And then I think patients also want that outlined verbally for them because sometimes reading the consent is really overwhelming. The other thing that is really important, and I always tell patients this, and I think it's really important for every patient to understand this. A consent form is not a contract. A consent form does not obligate a patient to anything. A consent form allows the physician to proceed with the clinical trial, but the patient can withdraw at any time for any reason or no reason whatsoever. And that is the basic ethics of clinical research, that there is never a time where a patient must proceed in any way. So I want to emphasize that here. And I do tell that to every patient because sometimes if you don't, they feel like they obligated themselves. And then the last thing I want to mention is it more likely for the cancer or the clinical trial to kill you? So the way we do clinical trials now, safety is really paramount. And even in the most experimental clinical trials, the death rate that is related to drug is generally in the range of about 1%. It is very low in cancer trials. There are many safety features that are built in. So for the type of patients that I see, most of them have a median survival of expected 9 months to a year. So 50% will have died of their cancer in 9 months to a year. And we can compare that to a 1% chance of dying due to the effects of drugs. Clinical trials are way different than they were, let's say, 40 years ago. And there are just so many safety features that are built into them. And these patients are followed extraordinarily carefully. So I did want to emphasize that.
Is that 1% related to the hyperreactive chance that you see in some patients you mentioned?
No, actually, the hyperreactions we saw in patients that got FDA approved drugs. We saw it, the drug was FDA approved, and we noticed that patient that we had, initially we saw 2 patients in clinic. And, I'm a pretty seasoned physician, and I'd never seen an acceleration like this. And then we began to look for more patients. But I do want to emphasize, and this is really important, so there is a small percentage of patients with immunotherapy that develop this accelerated progression. It's probably in the range of less than 10%, maybe less than 5%. Obviously, very important for those patients. But in spite of that, I will say that I think these immunotherapy drugs are the best drugs that have ever come on the market. And it's because of the larger percentage of patients that respond, and even more than the response. And I showed just a couple of patients like this, but we've had a lot that have very end-stage disease, and you give them immunotherapy, and the disease is, I think, cured. I mean, they go into complete remission. And so I do want to emphasize the hyperprogression. We're one of the groups that discovered it. It's a very important side effect. We think that there are molecular markers that increase the risk, but I don't want to diminish how important I think immunotherapy is, because I've seen so many patients that have had unimaginable responses that were unimaginable just a few years ago.
Yeah, by way of summary too, I go one step further that any clinical trial or research study that's going to involve human subjects, if you know that the risk of getting the treatment that is going to be tested is worse than the risk of whatever disease is being treated, I don't believe any human subjects committee would approve that study. So not just for this kind of research.
I agree.
Yeah. So we're running out of time. So room 6, you get to have the last summary from one of the groups. So
most of the things we discussed already being mentioned, but one that was also came up in our group, what other alternative treatments are there that our group wanted to know during the informed consent process?
Okay, so Razelle, I assume if somebody's come to you, it's because other alternative treatments simply have not worked. But is there anything else you have to offer them on that count?
Sometimes it's because no alternative treatments have worked, but sometimes there are alternative treatments, but they often have very low response rates. So they may have response rates of 5%. Or 10%, or the responses when they occur are known not to last long. So there has to be, by the people who review the clinical trial, a belief that what you're doing for whatever group of patients you're doing, that the chances are it's going to be better than the alternatives. But for an individual patient, we never know. Again, the clinical trial will not be approved if, from a statistical point of view, the belief is that the approved therapies or that there are approved options that would be better than the clinical trial. Now, of course, it could turn out that that's wrong, that the clinical trial doesn't work at all. But in general, you're pitting the clinical trial against something with very low response rates, 5% or something like that.
Okay. Well, we've remarkably already reached the end time for this evening's program. Razelle, I want to thank you for another really interesting talk and presentation. You covered a lot of very difficult ground, and I think did it in a way that everybody could understand. Much appreciated. I want to thank the participants this evening for your comments, and I want to encourage everybody to keep these conversations going. Talk to your friends and family about what you heard today, not just because of the science, but because we want the science to be done well. So we need to talk about the ethical issues. So thank you, and everybody have a good evening.